Good morning. My name is Joe Komsky, and I'm the Senior Director of Philanthropy here at Sanford Burnham Prebys. You may remember me from our Insights lecture series, but today we have a very special presentation for you on rare childhood diseases with two of our leading scientists and two of their patients that they've been able to help. Now, we know adults get sick for a variety of reasons, sometimes of our own making, but sick children are truly innocent. They tug at our hearts, and we want them to get better. Sometimes it's a passing illness, easily treated with medicines, but others result from inherited conditions, and their effects can last a lifetime. Many of these diseases are extremely rare, afflicting a small percentage of the population. But to those who have the disease, the effects can be devastating. Sometimes parents don't even know why their children are sick, and they go on a years-long diagnostic journey. Thanks to DNA sequencing over the last 20-some years, scientists have identified the causes of some of these disorders. But even so, it's only a fraction of the diseases out there. Diagnosing these conditions is just the first step in a family's journey. In most cases, the family doctor has never encountered the disease before. Then the great majority of these genetic diseases have no treatment, leaving families bewildered and afraid. Today, two of our scientists will tell us about how they helped families in these exact situations. First, Dr. Hudson Freeze is the director of the Human Genetics Program at the Sanford Children's Health Research Center. He's been at Sanford Burnham Prebys for 33 years and worked on genetic disorders in glycosylation. Sorry, I still haven't pronounced it right. Glycosylation. Hud, a lot of people may not know what glycosylation is, and we'd love for you to be able to explain it to us and tell us about your research and how you were able to help.
Well, thanks very much, Joe, for that nice introduction. Yeah, I work in a strange area of science called glycobiology, and that's not an area that most physicians and sadly even most scientists don't really know and understand. But you know, it turns out that there are patients out there who have defects in this process of glycosylation, which is adding sugar chains onto proteins. And there's lots of different ways to make mistakes, lots of different ways to impair that really important situation. So we have been working in these congenital disorders of glycosylation now for, well over 25 years. And in that time, we've been very fortunate. We've been able to diagnose probably about 360 patients, discover or co-discover probably 25 different disorders. So, you know, we've been at the business for a long time. And I think that our approach has been kind of unique. And I think the story that I'm going to be able to tell may explain a little bit about that. Okay, so I am going to share my screen. And I'd like to tell you about what we call the Cdg ecosystem. And it's really symbiosis at work. And you all know about the connection between physicians and scientists, and we exchange information and we're on the same page. Well, not always, and probably not enough. We need to exchange more information. We need to have a closer, intimate relationship. But the trainings are different, and I'm not trained as a physician. I'm a basic scientist. My area was glycobiology, so I didn't know very much medicine. However, I was involved for quite a while in doing some things. And as you know, the main thing that you have to be able to do is to work with the most important aspect of this, which is really the patients, the families. Those are the things that we as researchers oftentimes leave out because that's not part of what we do. And of course, the main connection is between the physicians and the families. But because glycobiology wasn't very well known for a long time, families desperate for finding out what could be wrong with their kids would then turn to us. So that completed the circle. We were able to be part of the care in some ways. Of course, the physicians are primary ones, but we were able to become involved. And we think if we're going to do that, we owe something back to the families. And so where we could do that, we provided information. Sometimes it was diagnosis. that then would be authenticated by their doc. But it was the kind of thing that got us going in the morning, because it wasn't just us doing lab experiments, it was about the families. So what are rare diseases? Well, they're defined as any disease in the US under 200,000 patients. And most of these, as you can see at the bottom of the screen, most of them are rare recessive disorders. That means they're inherited in a way that requires 2 damaged genes, one from each parent. So if you look at this little cartoon above here, dad and mom are quite happy they don't have any disorder, but each one of them has one mistake in a gene. Not every gene, just some genes. So if dad has a mistake, mom has a mistake, when those gametes are produced, the sperm and the egg, those 2 separate. So here's a good gene going there, there's a damaged gene going there. The same thing is true for eggs. Now, when the children come along, they have a 1 in 4 chance of getting that double mistake, one from mom, one from dad. And so this is typical of what we find in a lot of these disorders, but there are other types, but this is the majority. And so when you look at what's gone on over the, I suppose about the last 40 years, you can see that very early on, back in 1981, when I first worked on a thing called eye cell disease, that was the first kind of congenital disorder of glycosylation that we could track down. And it's just grown enormously over the years. And that's a lot due to genomic sequencing and exome sequencing. Those are terms you probably hear. These different colors are just there to indicate different pathways that glycobiologists sort of categorize things into. And because we're so connected to the patients and the physicians, we were able to, over the last 10 years, organize meetings. And we have, here is a picture from a couple of years ago when we had a meeting in San Diego from Cdg Care, which is the family representative organization, and ngly1.org. That was another, and now they're continuous as a single organization. So you can see that there are a lot of patients, a lot of people that we've gotten to know pretty well. So I want to talk about one rare disease. It's a recessive one, and it's called CAD. Now, we don't need to go into what all that means, but there's some characteristics that you need to understand. Again, it goes back to that same cartoon that we drew a little bit earlier. So 1 in 4 children could be affected by it. Well, CAD makes uridine. That's an important molecule for the cell, but it can make it from stitching together other molecules. And the problem is the CAD gene is very large. It is so large, in fact, that there are probably 1,000 different variants, 1,000 different variants that could be potentially damaging, but we don't know which ones are damaging. So our lab was the first one to work out and to discover this disorder. And that was great. And we had a little cell system we put together. We knew how to arrange this in a way that would actually take care of diagnosing whether this was a damaging effect. So the important thing out of this is that uridine was capable of correcting the defect. Now, how do you do that? Well, we don't only make uridine, we can also use it in our diet. So if you give uridine as a supplement, that's one way you can make this better. So we realized that we could rescue cells, and that was great because about 24 physicians then said, well, I have patients who look like that and they have these problems. And I found from exome sequencing that they have a couple variants. My gosh, maybe they're really CAD patients. And maybe they can be rescued with uridine. We realized, though, that we needed a more sensitive test than the one we had. It wasn't quite good enough. So what are you going to do when you don't have the right test? You just hope that you can come up with one, or as biology and medicine works these days, everybody around the world can have the opportunity to communicate. And we met with and collaborated with Santiago Ramon in Spain, Valencia now. He's a CAD guru. He knows CAD inside out. And he had a better way to do an assay. So we could take all of those patients, all those 24 patients, and now we could test them in his system and see if they really were patients. And you know what? Only 10 of those turned out to be real patients. Why was that important? Well, think if you're a parent and you want to be assured that uridine might help your child. We can tell with this test, and that's why it's so important. So we were able to very fortunately meet Dr. Kristen Lindstrom and Matt Traster from Phoenix Children's Hospital, who introduced us to Audrey. And that was just a year ago. So this will give you a bit of an idea of the situation.
So we're on day 2 of the uridine. We haven't given her.
So you can tell that Audrey is bedridden. She doesn't make purposeful motions. She has lots of seizures. And then here is 6 months later after being on uridine.
Yeah.
What is that?
Well, that's your book.
I don't need to say anything. You see it for yourself. Let's meet Audrey and Jessica, her mom. Okay.
Hello.
Hi.
Hi.
Hi, Jessica. Audrey, hi.
You're here with us. Ah.
Now I can see you a lot better, Audrey.
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 Say hi.
Whoa. You're on television. There are lots and lots of people looking at you. Look at you.
Look at you. You say hi.
So, Jessica, can you tell your story?
Sure. Yeah. Well, we are obviously just so thankful for your team, Dr. Lindstrom, Audrey's geneticist, for introducing us to you guys and your research, and then Audrey's neurologist, Dr. Traster. If we didn't have this incredible team, Audrey would not be alive and thriving like she is today. So I just want to say that from the offset, that we are so incredibly grateful. So I guess I'll start at the beginning. Audrey was a healthy 6-pound, 8-ounce baby 9.5 years ago. Pretty early on, we noticed she was delayed. She didn't roll over until she was 9 months, and she was globally delayed in other milestones. So we knew that pretty early on. Therapy services, we started early on, and then she went to a special needs preschool at 3 years old. When she was 3, when she was 3 years old, Audrey had her first seizure. At onset, it was caused by a fever, so we dismissed it as a febrile seizure. We went on with our lives for the moment, thinking it was just a one-off. The following year, Audrey's life took a dramatic turn. In March 2015, she was officially diagnosed with epilepsy after having 11 focal tonic-clonic seizures in a day. She would desat every single time, depriving her brain of oxygen. We started off on Keppra, and we're hoping that seizures would be controlled, and they weren't, which then began our incredibly disappointing journey with pharmaceuticals. She was then put on Lamictal and Trileptal. Both were incredibly harmful to her. Mr. Abby. And then she began having 25 to 45-minute tonic-clonic seizures once per week. We were calling 911 once a week for months during that time, to the point where the paramedics started recognizing Audrey as a regular. She stopped talking. Her progress towards her goals in school just plummeted. She developed tics on top of the seizures. We stopped those meds and began our research into cannabis. Here in Arizona, you can get a medical marijuana card for children with life-threatening conditions. We explored that route. We prayed that marijuana was the miracle plant for us, and it did help for a time. Her seizures weren't as severe, but as with her other meds, we didn't get seizure control, and she was still having a focal or tonic-clonic seizure once a week or every other week. In 2017, we did an epilepsy genetics panel. We were convinced she had Dravet syndrome. We were convinced she had Dravet syndrome because she had all the symptoms for that syndrome. She was negative for that gene, and her test results came back inconclusive. We were relieved she didn't have Dravet, but we still just came up empty on what is going on. We came to the conclusion that we'll likely never know what was causing Audrey's seizures. Yeah. And basically this was as good it was gonna get. This is it. You know, she's gonna have a tonic-clonic seizures for the rest of her life. And it just is what it is. It was our new normal. Audrey is never allowed to be alone, or was never allowed to be alone, including at night, because she would often seize in the middle of the night. So we were just constantly on high alert. Now, introduced 2019, Audrey took another downturn. Her seizures became more severe. When she would have a focal or a tonic-clonic seizure, she would turn blue almost every single time. Her oxygen saturations would go into 10, 30%. She would code sometimes and just completely stop breathing. In the summer of 2019, she basically stopped walking. She constantly required a stroller or a gait belt to just get around. She was a shell of who she was. We started looking into VNS. She had a surgery for the VNS. August of 2019, she's currently on the highest possible settings for that device. There was still no perfect solution and no tangible sign that it really made a difference. And in December 2019, our worst fear was realized. Audrey went into status epilepticus. She was seizing every hour and destatting every single time. They gave her Ativan, which in the past often worked. It did not work this time. She eventually had to be intubated and put in an induced coma to give her brain a break. She remained in the hospital for 21 days in the PICU. She was on ketamine, phenobarbital, and Depakote during that time, and she was still seizing several times a day, even with those really intense meds. We were discharged after 21 days with very little hope. As you saw with Dr. Freeze's videos, we have tons more of our own. Audrey was immobile, nonverbal, and basically catch. Are you playing catch? I'm trying to do this. Hello? Hello? This is like the intense part. Yeah. So she was immobile, nonverbal, and basically in a minimally conscious state. We did everything we could to help her rehab, but nothing seemed to work. She was barely alive and still seizing frequently. And we were just seeing deteriorate before our eyes, and we couldn't do anything about it. It was just heartbreaking. Dr. Lindstrom, who was Audrey's fabulous geneticist at the time, gave a more extensive genetic testing. And while Audrey was in the hospital, she knew how desperate we were and wanted to help us in any way she could. She discovered that Audrey had this Cdg disorder, which explained all of Audrey's symptoms from the delays to her seizures, and is, as we already know, extremely rare. Now, oftentimes, at least how I thought about it, oftentimes with genetics, that's it. Like, you know the cause, but there's really nothing you could do about it. Now, Sorry, I lost my place.
You know,
Now,
and most rare disorders are not treatable.
no, and so that's how we
90% are
took
not
it.
treatable, even if you know what they are.
Exactly. And so that's how we took in the info of like, okay, she has this incredibly rare genetic disorder, now what? And she then told us that with the help of you, that uridine was a possible solution. And that just gave us the slightest glimmer of hope. Now this is, you know, after we've tried VNS, medical marijuana, meds, keto, like diet changes. We tried pretty much everything in the book. And so we had a little glimmer of hope, but not much because we were pretty worn down at the time. We put Audrey on uridine after the help of Dr. Fries and Dr. Lindstrom on February 4th of 2020. We saw results within a week. She started vocalizing again. She began moving. And for the first time in months, she stopped seizing. We couldn't believe it. For the last 5 years, we did not know what living without a focal or tonic-clonic seizure was like. And February 10th, 2020 was her last tonic-clonic seizure. Within a week of starting uridine, she could lift up her head. Within 2 weeks, she was trying to sit up and stand up. Within 3 weeks, she was laughing and talking and dancing and reacting to music. She loves music. And so she started reacting to all of her favorite pop stars. Within a month, she was able to start eating solid foods and not need to be tube fed. The miracles just kept coming in. In June of 2020, she began walking again. We are still constantly amazed at the progress Audrey has made in such a short amount of time. She's achieving milestones we didn't even think possible. We're starting potty training. We never even thought that that would be possible. We were in literally a life and death situation with Audrey, and her quick turnaround is nothing short of a miracle. We are so, so grateful for the help and research of Dr. Fries and his team and those here on our team in Arizona to save Audrey's life. We're just indebted to you guys. Thank you so much.
Wow, what a story. What a story.
And
to see her to see her now, you know, that's why we do what we do.
Yeah.
Because I mean, this doesn't happen very often.
No.
And there are other children who've been treated with uridine and a modified form of it, which was actually the one you used.
Yeah.
But you know, and that's been pretty successful in those individuals as well. So you know, I think what you do is you show us, you inspire us. We come into the lab and we see Audrey's picture.
Yeah.
You know, and that says that's who you're working for. That's what's going to make the difference here.
Yeah.
So I can't thank you enough for telling your story.
Well, I can't thank you guys enough.
And we're going to turn it back to Joe here.
Okay.
Thanks again.
For those of you in the audience, if you have questions, there's a Q&A button at the bottom of your screen. We have a couple of those that have come up. I was going to say first for Jessica and Audrey, normally we ask our speakers to make sure that there is no background noise. Never have I been happier to hear all the background noise that we heard while you were speaking. That is absolutely amazing. And we actually had a comment come up where someone said, I just want to say amazing. It is absolutely tremendous. One of the questions that has come up already is, has the uridine cleared up her seizure activity completely?
No. So yes and no. So she has a lot of seizures, like a lot of types of seizures. So tonic, clonic, and focals were the most devastating and life-threatening ones. And she also has absent and myoclonic astatic seizures. So she still has those. So those are like little nanosecond per day. And so she still has those, but we're still kind of, it's just been a year. So we like don't know what like the future holds, like with like being able to, you know, get a control of those ones. But we're, I mean, and we haven't needed like medical rescue meds in over a year. So we were doing midazolam, diastat, clonazepam, all the PAMs back a year ago, and we have not needed to use those for quite some time. So yeah.
Great. We have a couple other questions here that we'll answer live. HUD, for you, the question is, should CDG parents get the COVID vaccine? And if so, is one better than another for that? Do you have an opinion on that?
Yeah, well, first of all, I'm not a doc, so this is not medical advice, but at least some of the chatter that's been going on, you know, among the CDG groups is that in general, there's not a downside to getting the vaccine for CDG patients. So as far as which vaccine would be better, get what you can get. I don't think at this point, you know, we can be too choosy.
Okay. Then another question, someone typed in that their son has PMM2 CDG and asked, is there a similar treatment available for that subtype?
The answer to that is no, nothing that looks so miraculous, but I can tell you that there are different kinds of therapies that are being worked on now. You know, there's nothing that's sort of ready to go into the clinic that makes a difference in that. But there was a study that was done in Spain, and now that's being replicated here in the States for something that helps control seizures. So that is ongoing and underway. There are potential other drugs that may have quite an amazing effect, but that's in very, very early development. But we're really hopeful. But there's nothing out there that you can go take right now.
Let's finish up two things here. I want to finish up with two more questions. This next one will be for Jessica. For those of you, if we don't get to your questions here live, we will email them to the doctors and they will be able to answer them via email. So Jessica, the question again was about the developmental milestones and which developmental milestones is she starting to hit and which kind of maybe are on the horizon that you're looking forward to?
Well, again, I mentioned potty training. We never thought that we would even be close to being able to do that previously, because just like cognitively, I don't know if she would be able to understand that, but she's now like learning. We're working on that. Also, like just affection, like just social awareness of her people when she was so cloudy because of the seizure activity, I'm sure brain trauma, and the amount of meds that she was on, being able to be socially aware of others, even like her surroundings, she like was not, just like very foggy, I guess. And she's singing songs before she was singing like the Frozen song, like Into the Unknown. And so just more aware of her social surroundings. She's able to retain information more. So before she would do an OT skill or a PT skill with her therapist and she would lose it immediately because she would have a tonic-clonic seizure or whatever in that week. And it was 1 step forward, 3 steps back, 1 step forward, 3 steps back. And so, you know, being able to walk, her trunk control, it was very weak a year ago, and she has more. I mean, her PT every week is like, oh my gosh, she's like sitting up from the peanut ball and like barely needs assistance. Like she wasn't even doing that on the trampoline. Yeah, on our outdoor trampoline. Like she wasn't even able to do that before. standing in the pool on the shallow end, like the coordination that takes, she's able to do that. Verbalizing her emotions. She'll tell us when she's frustrated. Well, before she would just, but now she's able to verbalize her frustrations and recognize that when she, bothering her brother, say, and he'll say, oh, he's frustrated, he's upset right now. And I'm like, that's right. Yeah, you knocked over his blocks. So just like the social awareness and being able to vocalize those things, we just never had that before. She's so that's like huge, like for us in our minds. There's like tons more. Pretend play, she's like playing with her Abby doll right now. Sorry, there's like, I could go on.
We do have others who are going to jump on that we would love to hear it.
No problem.
Let me ask one last question and also let people know, HUD, I know you're typing some answers in because I can see it here on the controls, but we will also get back to some of these questions at the end. But HUD, what's on the horizon now? What are the next steps in your research at this point? What are you working on?
Yeah. So with CAD, as I mentioned, the only place you can actually have this assay done that will let you know if you have 2 mutations is in Spain. It's a fairly complicated assay, but what we want to do is to set it up here in the US. And we're really fortunate because the NIH has funded a 10-center consortium of different laboratories and hospitals, physicians who are studying various kinds of CDG. And so we're going to use some of that to go forward in that area and set that test up here. The other thing is that, as you saw from the growth curve, there are lots of CDGs that have been discovered, and we've got 2 more in the pipeline that we're going to be working on over the next year. So we're going to continue working on this, try to optimize those things. Plus, we have other projects involving different types of CDG that we're working on. And so we're excited, finding really quite amazing things.
Dr. Fries, thank you so much. Jessica and Audrey, thank you. It's an amazing story. But now we're going to hear from another researcher and another amazing story from one of his patients. Dr. Jose Luis Milan is a world-renowned researcher and professor in the Sanford Children's Health Research Center. He is also on the scientific board of Soft Bones Incorporated, an advocacy organization dedicated to people living with hypophosphatasia. Dr. Milan, we'd love for you to tell us about your research, what's on the horizon, and introduce us to your wonderful friends.
Thank you very much, Joe, for that introduction. My laboratory has spent pretty much the last 4 decades studying molecules that participate in the initiation of skeletal and dental mineralization. The studies for many years were very basic, try to understand how these enzymes all work together to bring calcium and phosphate together to initiate calcification. This is what is called basic research. But this all changed in 2004. Let me see if I can bring my screen up here. When I was invited to join the scientific advisory board of Hypophosphatasia Europe. These 2 individuals, Gerard Brandt in Würzburg, Germany, and Steve Ursprung in the region of Alsace in France, invited me to join their board because they were aware of the work with the animal models of hypophosphatasia that we had developed. And they were forming the very first organization of this kind to really bring support to the few patients with hypophosphatasia that were asking for help. There was little known about this disease. There were few people to turn to for support, for information. And they were pioneers in this sense, building up this organization. And I remember them asking me, what shall we do as a patient organization? And my answer at the time, half jokingly, was, let's bring everybody that can pronounce hypophosphatasia together to the same room. Let's see what the state of the art is. Let's see what we know scientifically and where we could take it from there. What we knew was the following. We knew that this severe lethal form of hypophosphatasia, where these children barely mineralize some of their bones, was very well mimicked by an animal model of infantile hypophosphatasia that we had developed. And this paucity of mineralization, this lack of mineralization, was caused by accumulation of inorganic pyrophosphate. Inorganic pyrophosphate has been called the water softener of the body. Essentially, it prevents ectopic, unwanted mineralization in all the soft tissues. However, in the skeleton and in teeth, there is an enzyme, alkaline phosphatase, that normally hydrolyzes, destroys this pyrophosphate and allows calcification to proceed. Here you see this black mineral, that it is the way it normally appears under the electron microscope, but in the mouse model of hypophosphatasia, there is really no calcification to speak of. The collagenous matrix is essentially uncalcified. So that we knew. What we did not know was how could we bring the missing enzyme to the sites of calcification in order to correct the defect. Now, shortly after, I was invited to a meeting in Montreal, Canada, by a small company, Enovia Pharma. They essentially gathered together what they call the dream team of alkaline phosphatase. Here you see Michael White, the world's expert on hypophosphatasia. You see Graham Russell, the co-discoverer of bisphosphonates based on the structure of pyrophosphate. Clark Anderson, who discovered matrix vesicles that are essential in the initiation of mineralization. My lab, that had an animal model that would be suitable to test a new mechanistic principle. They thought that they knew how to target alkaline phosphatase to these sites of mineralization by modifying the enzyme with the mineral targeting motif. They produced the enzyme, they sent it to my laboratory, and everything worked just beautifully. The mice were essentially completely treated. They had no further manifestations of the disease. And then the meeting that Hypophosphatasia Europe had planned for came about in May of 2007 in Alsace. These were all the people that were invited to the meeting. The meeting was hosted by patients and their families. They cooked for us. They shared the meals with us. They attended all the lectures. It was really a very family-oriented gathering of physicians, of research scientists, of patients. And it was at that meeting that I presented this preclinical work. And it was at this meeting that the clinical trials for hypophosphatasia got organized and initiated. Here I show you a couple of videos of two of the patients that were engaged. One was baby Dania, who in the United Arab Emirates was a 3-year-old girl, was the oldest patient to be admitted to the clinical trials. She couldn't really go up the steps. She was breathing through a tube. She needed positive ventilation. And then I had the pleasure of visiting her one year later in Dubai, or in Al Ain rather. And she was walking on her own, breathing on her own. It was really a remarkable recovery for this baby girl. One of the rare disease symposia that we normally held at our institute, we got to see Evie here in a stroller, looking at a mouse model of hypophosphatasia that one of my postdoctoral fellows was showing her. And then Lindsay, her mother, sent me this video 4 months later after treatment. What a tremendous improvement in her quality of life already with just this short treatment. So this next slide, let's see if I can advance it, summarizes this journey, which has been really quite remarkable, from concept design of the biologic, of the therapeutic, to preclinical testing in my laboratory, to this meeting in Unang, France, where essentially we had the world's experts coming together. Michael White, who was in charge of the clinical trials, Cheryl Rockman Greenberg, the first physician to treat a patient with HPP with this enzyme replacement in Canada, in Winnipeg, very appropriately so, because hypophosphatasia was first described in Canada. So it was really very nice that the first treatment could be realized in Canada as well. And this is the efficacy. You see the same hand you saw before of this baby affected by severe form of hypophosphatasia, the dramatic improvement after 24 weeks of treatment. Well, this treatment was then approved in 2015 for pediatric onset HPP. what this slide also shows is why this journey was so successful, because you essentially had all the components in place ready to interact for the symbiosis like Hudson Fries was talking about. We had a preclinical laboratory, we had a small pharma company ready to move on a new concept. The advocacy groups and expert clinicians all coming together to develop this treatment. I continue to interact with these advocacy groups. Here is Soft Bones US, which started as an organization in 2009. I had the honor of hosting one of the yearly meetings in La Jolla in 2014. There is a sister organization in Canada as well, whom I have visited as well. And I also had a chance to host them again in 2019. These meetings are quite fundamentally important also for me to be part of, because I keep on learning from the patients what works, what does not work, what do we still need to research in using our animal models, how can we ask questions that could continue to be clinically relevant. This past year, one of my postdoctoral fellows, Flavia Amadeu de Oliveira, received one of the grants, Soft Bones grants every year, to help in her studies with gene therapy of hypophosphatemia, try to develop a second treatment or new treatment options for this disease, so that we can continue to treat not only the infantile forms, but also the adult and odonto forms of the disease. One of the poster children of hypophosphatasia is Morgan Fisher. She lives here in the area, and I've had the pleasure of meeting her in 2010 when she was just about to start her treatment. I could lift her in my arms then, and now I can no longer do so. This is the latest picture that I took, that was taken in her house in 2018. And I would really like for you to now listen from her directly about her story. So if you are there, Morgan and Kate, I'd like to introduce the two of you and thank you for making time to be with us this morning.
Good morning. Thank you.
Hi.
How are you,
I'm Morgan.
Good, good. So I really would love for the audience to hear from you. How, as a young girl, how did you experience your disease? And now as a teenager, that essentially has recovered to a large degree from the skeletal symptoms of hypophosphatasia. How do you see what will become a lifelong treatment for your condition? Tell us your story, please.
Okay. I'm Morgan, and this is my mom, Kate.
Good morning.
And I just want to thank you, Dr. Milan, for your dedication to hypophosphatasia and for helping finding a treatment. And when I was born, my parents were told that I would never walk and that I would always be in a wheelchair and my quality of life would be very difficult. And so I met Dr. Milan when I was 5 years old. And then soon after I started my treatment in Canada with Dr. Greenberg. And we also met Dr. White and well, when I first met Dr. Milan, we went to his lab and we saw the mouse model of hypophosphatasia. And it was very fascinating to me to see like the mouse that had what I have. And as just as interesting as it was for me to see the mouse, I'm sure it was just even more fascinating for Dr. Milan to see me and for him to ask questions about how I felt and my symptoms that he can't ask a mouse. And after I started my treatment in Canada with Dr. Greenberg, a month after I started, I was the first time I ever walked up a staircase. And that was amazing because I had never like
Unassisted.
yeah, unassisted. And
That was about a month into treatment. Morgan is, I'm trying to let her talk because I know people want to hear her and not me. So I know that Dr. Milan, Morgan and Dr. Milan have been friends for quite some time, almost over a decade now. And no, I think you're about the same height now, right, Dr. Milan? But Morgan has was when she was accepted into the trials. We traveled back and forth to Winnipeg, brutal 24-hour, 48-hour trips to see Dr. Greenberg and do numerous tests and lots of blood work. And the Strenzig has literally changed her life, without a doubt.
And without Strenzig, I wouldn't even be able to walk or do anything I can now. And I can even ski, and I can ride my bike and walk my dog down the street. And that's just, I wouldn't be able to do that without having this treatment.
Wonderful, wonderful.
I mean, we could go from, birth to now, but I don't know if everybody really wants to hear that. But Morgan did break all of her extremities in utero, which is when we actually realized that she had this rare disease, and hasn't broken a bone, thank goodness, for 15 years. However, recently she just dislocated her elbow and took a little chip out of her humerus. So that's the first time that she's ever broken anything. And she had a pretty challenging 2020, just like the rest of us. She did battle Hodgkin's lymphoma. And so I believe with chemotherapy, her bones definitely took a hit. So she just recently had a DEXA scan done, and her bone results, it didn't come back as well as we would have liked it to be. But again, I think without Strenzig, that would have been much harsher treatment for her than a normal person going through it. She's in high school now, which is amazing.
And after I finish high school, I'm at high school virtually. And after I finish high school, I want to go to college and maybe study veterinary science or become a lawyer, because like my dad is a lawyer and he wants me to take over the family business. And I don't know, I'm pretty logical and I like to argue with everyone, so it's a possibility.
I do believe that, with knowing Dr. Milan and having been in her lab, in the lab, and meeting his team, Morgan is definitely much more interested in science. She loves that research. She's very interested in microbiology and the ocean, and so she's constantly learning something else and something new. So we're internally grateful to you, Dr. Milan, for dedicating your career to hypophosphastasia. And we do joke that, eventually we hope that Dr. Milan will make it an oral pill or a vaccine so that we don't have to do the weekly injections. Morgan has asked you that numerous times, I know. So you think she's kidding, but she's not. So, the hope, go ahead, Joe.
Well, I was going to say that's actually a wonderful point, and a question for Jose Luis as to what is next on the horizon for this now.
Well, that's precisely quite an important point, because first of all, this treatment, which is actually life-saving for children with the most severe forms of the disease, requires 3 to 6 injections weekly for the rest of their lives. There are injection site reactions, which sometimes lead to discontinuation of treatment because the reactions are quite severe. This treatment is also extremely expensive. So patients with the adult forms, the milder forms of the disease, adult and odontohypophosphatasia, do not have access to the treatment, nor would it be recommended, because it is just unaffordable for the milder forms of the disease. So we are trying to develop alternative treatments. In fact, gene therapy works quite well. A single injection in mice cures them for life of the mouse. So gene therapy is possibly a second-generation treatment that is most advanced. Together with collaborators in Japan, we have published several papers already, more coming out of my laboratory at the moment. So that is a second generation. There are other therapeutic principles that we're also exploring, 2-3 other principles. And those will come online as time goes by. We're researching on those. So indeed, I hope that to fulfill Morgan's wishes to either have a pill or at least one injection, not 6 injections a week for the rest of her life.
Would that be something where tests could be done either in utero or at birth, where you could identify somebody with the disease and gene therapy could start very early?
Yes, so the tests are already being done. You can get diagnosed if your fetus is affected during pregnancy, and gene therapy indeed has the potential to treat even fetuses with HPP. In mice, we have shown that that is possible. So again, it's a matter of translating this to humans, but that is where we're going now. Most of our research is now translational, trying to really help the patients that now are where what we know is not yet fixable. So.
Kate and Morgan, there's a question here for you about, was Morgan ever diagnosed with osteogenesis?
They definitely thought that when I was pregnant. That definitely was the main condition that they diagnosed her with. However, I did have an amniocentesis that we then sent off to the hospital in LA, and now I'm drawing a blank, but they were the first ones who actually did say hypophosphastasia. And I have to tell you, it was not easy finding a doctor that even could pronounce hypophosphastasia,
Even
let
now.
alone, yeah, even today. I mean, they turn it into hypophosphatemia, which is different, and or osteogenesis imperfecta. But her bones are much more pliable than they are brittle.
For my own curiosity, and I think for our audience curiosity, Morgan, what's your favorite activity right now? I want to hear how you're living life that you wouldn't have had otherwise. What do you love to do?
I do love to ride horses. That's my favorite. And I know it seems pretty like a little dangerous for me, but it actually is. It's therapeutic and it helps with my legs. That makes me like more more flexible and like that I can stretch. And so something, if I fall down, I won't break my hip or something.
That's wonderful.
She's not a big sports person. We definitely avoided the high impact sports and anything like that. So she's a swimmer, but she's also an artist. She plays the piano, she plays the bass guitar, and she's just picked up playing the cello. Now with a broken arm, that's been kind of difficult, but she's going to get back into it pretty quick here, but she's quite a musician.
But now that broken arm is just a typical teenage broken arm and not something much more serious, right?
Yeah.
Yeah, you continue to be an inspiration to me, Morgan. Thank you so much. You're my hero.
Thank you.
Back at you.
We're going to bring back our other guests, Dr. Fries and Jessica and Audrey. We have been blessed today with so many heroes, our scientists, the doctors, the parents, the children. This is truly amazing. Jessica and Kate, real quick, Jessica, we'll start with you. What was the, it must have been such an amazing sense of relief to even just know what was going on from this. Can you share that with us for a moment?
Yes. Well, as I said with Dr. Fries, I mean, there's 3 ways genetic disorders can be looked at. It's, you know the cause and you can't do anything about it. You know the cause, and there is something that you could do, or you know the cause and it's life-threatening and like they're gonna, oh, you're wearing a princess dress. Look at you.
You're a princess.
You are a princess. And so for her to be able to understand that she has a rare genetic disorder, extremely rare, and then we still, and the rarest of the rare genetic disorders, and then for us to have a solution to that through uridine, that was mind-blowing. Like we did not, again, we tried all the different everything. And so we gave up of like, okay, this is our life now. This is our new normal with her. And we didn't know what her life expectancy was going to be, her quality of life, anything. We just, at this point, just wanted to keep her as comfortable as we could. So it was insane that we could take a breath and not have to think about calling 911 all the time, or having to go to the hospital and get Ativan and all this stuff. And so it was, it was, like I said, just miraculous. It was just shocking and miraculous. Right?
Kate, how about yourself?
Same thing. I mean, before Morgan was born, they actually told us to abort the pregnancy because Morgan would be bedridden. Her quality of life would have been horrible. And there wasn't any treatment. So even when I talked to Dr. Milan, when she was 4, I just called him out of the blue, and he kind of directed us towards Dr. White. And then Dr. White directed us to Dr. Greenberg. So, you know, at 5 years old, Morgan didn't even walk until she was 3. And that was only because her younger brother was starting to walk and she didn't like it. But it was, and still is, it's very difficult to find treatment, not treatment, but a doctor that's willing to learn about HPP. And, you know, thank God for soft bones. And I reached out to other parents. Deb Fowler is she is a best friend, and we're sisters from different, you know, parents. Her son is exactly the same age as Morgan, and to be able to compare notes, because I'm always, if something comes up, I'll shoot an email off to Deb, and Deb gets me the answers. So, you know, it's extremely important to have a community too, and to have other people to know that we're not the only ones out there. So it's frustrating, but at least now we have a treatment, and Morgan is thriving, and she has a future, and she's going places. I mean, she's here for a reason.
Absolutely, absolutely. Jose Luis and HUD, I guess part of what you need to do also is help educate doctors and physicians and primary care physicians on these diseases. Obviously, there's wonderful groups like Soft Bones and HUD. Hopefully, you will mention some of the other ones for the diseases that you deal with. But Jose Luis, we'll start with you. How do you sort of do that? How do you get that wording?
Well, in the past, rare diseases were not only rare, but really people were not paying attention to them. Now they are in vogue, and people realize how these essentially experiments of nature are so informative about not only the basic biology, but that they affect real people. There are more than 7,000 rare diseases, so together they are not that rare. They're actually quite common if you pull them. We do our education in meetings where we present our research, but I think that the fabulous role that these patient advocacy groups play is just fundamentally important, and this is why I'm so delighted to be part of this advocacy group organization.
HUD, how about for you? What do you see as the important ways we can do that?
Yeah, I think that without the family groups, this doesn't go anywhere. And I've always said, and I always tell them, you're the experts. Mm-hmm. You live with this every day. I mean, we're in the lab doing our experiments, and what we see in patients, that, you know, of course, helps inform the things we think about, those things we go after, models that we might try to make. But I can tell you for sure that mom power is probably one of the most potent forces in the universe. And I have witnessed that so often. And in fact, I mentioned this grant that included a number of different centers, different hospitals in the States. One of the requirements for even getting in the running for that is that you have a strong association with a parents organization, with an advocacy organization. And, from my standpoint, I've known that for a long, long time. And now at this point, we're all stakeholders, we're all communicating with each other. And sometimes the questions that come to me, and I'm sure for Jose as well, that we get them, and we oftentimes will go back to the parents group and say, what are you guys doing?
So true.
And of course, with email and Twitter and everything else, the communication is just going on all the time. So we couldn't do it without them, bottom line. And CDG Care is the one that has galvanized all the glycosylation disorders in the US. There's another set of organizations in Europe, and we ally with them as well as in Canada.
Well, I love your comment about the mom power, and we have two beautiful examples of that in Jessica and Kate. That is, again, you are heroes to your children, you're heroes to us. Morgan and Audrey, you are heroes to us for sure. And Audrey, you are a princess. And Morgan, you're going to be a lawyer, and you can represent me anytime, because I am sure that you are going to be a phenomenal, phenomenal lawyer.
Thank you.
Dr. Milan, Dr. Fries, we want to thank you. You are heroes as well for the research you do on these rare diseases. For those of you watching, if we didn't get to your questions, like I said, we will forward them via email to Dr. Milan and Dr. Fries and our other panelists, the families if necessary, and we'll get back to you via email. We want to thank everybody for participating today. I hope you found this as meaningful as we did. If you would like more information about our research in childhood diseases or any of the research that we do, please feel free to call or email me. My information is on the screen. Again, we thank you so much, and everybody please have a wonderful day.